participants now. So, okay, well, welcome everyone. And this is the Mathematical Oncology subgroup keynote. And we're pleasure here to welcome Andrea Bild, who is not a mathematical oncologist. She's actually a biologist, an experimental one, and she works closely with mathematicians, as you're going to see. She got her PhD in Colorado, and that was in molecular oncology and pharmacology, and that was back in 2002. And then she moved to Duke, where she did a postdoc, and that was till 2006. And then she moved to a faculty position in Utah, where she stayed up until 2018.
Yeah, 3 years ago, yeah.
And she made the big leap over to City of Hope. And when we were discussing, I was trying to work out the first time I met Andrea, because she's been heavily involved in the Cancer Systems Biology Consortium, which is an NIH-funded consortium that I'm also part of. And we'd met at a few meetings over the years, and neither of us can quite remember the exact date, but it's probably been at least 5 years, give or take, right? But we're both heavily interested in tumor heterogeneity and the role that heterogeneity has in drug resistance and the feedback between context and heterogeneity. And so it makes a lot of sense that her Cancer Systems Biology grant, the big grant that she has, is with former SMB President Fred Adler. And both of them collaborate heavily on that. When I asked her for a sort of very high-level biography, she gave me 3 points. One was, she's a systems biologist, not a mathematician. The other is, I have lots of data and really appreciate and enjoy working with mathematicians. And I think that's to help understand that data better. And the focus is to understand tumor evolution and resistance states so that you can better treat patients. And so I think that aligns very much with many of us interested in mathematical oncology, which is, if we can understand how the disease evolves, we can potentially treat it better. And so we're very much looking forward to your talk, Andrea, and the title is very enticing, with competitive and cooperative interactions being there, because of course that's stuff that modelers absolutely adore, as I'm sure you're aware. So
yeah.
A pleasure to have you here, Andrea. Take it away.
Yes, thank you, Sandy and Heiko, and it's been a pleasure working with you all, as well as the lead mathematicians on this project, specifically Drs. Jason Griffiths and Fred Adler. And I'll also be showing the work of our Bild scientist, Rena Ewens, and our bioinformatician, Jinfeng Chen. So this is really a team effort, and I hope you enjoy the work that we're doing. So our lab really focuses on the evolution of cancer, and shown on the right is just a cartoon of how we think solid tumors evolve during therapy. So this plot shows a heterogeneous tumor where these different colors represent different clonal populations, which might have, for example, different mutations or different phenotypic characteristics. And as you can tell, as the tumor grows, these subclonal populations evolve. And during drug treatment, some of them go away, some of them evolve further to a more aggressive or resistant state, and ultimately leading to a refractory tumor to which the patients succumb. So our team's goal is really to understand how cancer evolves, and the relationship between these subclones is they evolve during therapy. And my work with the mathematicians on the team include integrating population dynamics and modeling with cell phenotypic evolution to study resistance, as well as how to reverse resistance. And what I'll be talking about today, the model system I'll be using is estrogen receptor breast cancer, which proliferation is driven by estrogen signaling to the estrogen receptor, causing proliferation of these cancer cells. And the 2 key objectives of the study I'll be discussing today are really to understand intracellular evolution. So in this case, that means as cancer cells are treated and over time, how do they evolve to resistant states, as well as how cells interact in the tumor microenvironment. So as we just showed in the prior figure, cancers are heterogeneous, and if you have multiple subclones within a tumor, Some might be resistant, some sensitive. How did these cells interact during the treatment and over time? So the clinical trial I'll be discussing today is focused really on a cell cycle inhibitor and endocrine therapy treatment. So I just wanted a brief review of cell cycle signaling. There's not many signaling figures, so don't worry too much. And in general, with cancer cells, they're not dividing, they're in G0. But as cells get proliferative signal, such as estrogen, they go through a growth phase, a G1 phase, leading to DNA synthesis, which then leads to G2M or mitosis and cell division and proliferation. And genes that are critical to this decision to go no-go are this cyclin-dependent kinases or CDK proteins shown here, which bind to cyclins. And the drug that we'll be talking about today, or the two drugs we'll be talking about today, is this drug called ribociclib, which is an inhibitor of this complex. So it blocks CDK4/6 activation and therefore blocks proliferation through the cell cycle, as well as letrozole, which is an inhibitor of estrogen receptor signaling. And it's well known that estrogen receptor also signals through this critical CDK cyclin pathway. And so you can imagine that in patients, when you have combined letrozole ribociclib treatment, you might more effectively block proliferation of their tumors. So that leads us to the clinical trial, which we'll be discussing in the first section of the presentation. And the clinical trial is called Feline. There's always cool names for clinical trials. And basically, this was a clinical trial in which estrogen receptor positive breast cancer patients were given either standard of care endocrine therapy, which is letrozole alone. So this is normally what women would get in the clinic. These are postmenopausal women. And the two other arms, the patients were given the standard of care therapy plus the CDK4/6 inhibitor, either at an intermittent high dose or a constant low dose. And really the goal of these different timing and dosing was to maximize response and minimize side effect profiles of the combination in this postmenopausal population. It wasn't really to do the modeling, but that was a great side effect of these three different arms. The clinical trial itself includes 121 total patients, and for each of those patients, they had 3 different biopsy times. The first time was prior to treatment, where we get a biopsy from the breast tumor before they're given therapy. And then we get a biopsy 14 days after therapy is initiated. And then lastly, we get a biopsy 6 months after the initiation of therapy at time of surgery. And so these biopsies allow us to perform genomic analyses to really understand the different genomic characteristics, the signaling characteristics and phenotype of these cells over time during the different therapy arms. Now, in order to measure response for these patients, these estrogen receptor positive patients have different modalities, and this includes a clinical assessment. This is basically calipers in the clinic that they measure the size. It's a pretty inaccurate way to measure a tumor size. We also have measurements of imaging, including ultrasound, mammogram, and MRI. And then last, we have surgical pathology that actually, from the dissected tumor at time of surgery, actually accounts for the true size of the tumor. And interesting, in ER-positive cancer, it's very difficult to measure accurate size of tumors as they're responding to therapy, in part because these tumors are slower growing and they're slower to respond as well. And so that was the first hurdle of our study, which I'll talk about in the first part of this presentation. Now, it's important just to say that from the biopsies, we are able to get both single cell RNA expression, which measures from each individual cell in the tumor, measures the phenotype of the cell, the signaling state of the cell, and so we can measure those states over time. We also get DNA sequencing, which measures the genetic changes in the tumor. And I won't be talking about this data today, but we have this as well if anyone's interested. Okay, so the outline for our presentation is, first, I'll discuss how we use this imaging data in a mathematical modeling to reconstruct and classify patient response to therapy. That's really the first step to be able to classify patients as responders or non-responders. And that annotation is then linked to our genomic data to better understand how tumor cells evolve during therapy in these two distinct populations, and to understand how these different drug timing and dose impacts cancer progression. And then last, I'll talk about a model system we have for ER-positive breast cancer, in which we test resistant traits and how to reverse them. in this system. Okay, so first we're talking about the modeling. So with imaging in breast cancer patients, there are a few limitations. One is that current methods in the clinic don't integrate size measurements. And so the clinicians, for example, are getting different measurements. Normally they don't get MRI for this population, but for the case of this trial, they are getting that measurement. But usually they do get these others, and they're not consolidated for an integrated measurement of response. And it's really complicated to measure true response to therapy for these women. The second issue, the second hurdle is that the images are taken intermittently. So you can imagine for a breast cancer patient, if she has imaging a few months apart from each other, this time point A and time point B, you could have a patient that actually responds initially and then rebounds. And this patient might look the same from these two time points as a patient with a stable response. Now, clinically, these are two very different patients. One is actually becoming resistant. One is still responding to therapy. But by a single imaging modality and two time points, you might not be able to see these trajectories. The second challenge that we have with the different imaging modalities and why they haven't been combined previously is that they have different biases and are relatively unreliable. And so here this shows one example of this, where you have the patient tumors from this clinical trial plotted on a graph for size, and you have the ultrasound measurements on the x-axis and the pathological measurement in the time of surgery on the y-axis. You can see as the tumors get larger, the ultrasound measurements are less accurate. So this is reflected by these tumors shown here. And so each imaging modality has its own unique flaws, and including those biases in the model will help us retain a more accurate measurement of response. So to build this model, Jason built a dynamic response evaluation model in which he first fit the curves using a Gaussian process. So he estimated the projection of the trajectories from the various different measurements. And an actual patient is shown here, where you can see the colored dots represent the different measurements that this patient had over the course of her treatment. And from that data, he then used a probabilistic model to account for the biases and variability of the different imaging methods. And so at the end of that, at the end of the model, the outcome is a trajectory of each patient over time that accounts for all the imaging metrics and accounts for uncertainty in the measurements. So shown in the top panel are the patients from each arm, either the letrozole or endocrine therapy alone, or the letrozole plus high-dose ribociclib, high-dose intermittent, or low-dose continuous. What you can see is that all three arms have both responder patients. Those are the patients whose tumor decreases in size, those patients in red shown here. And all arms also have non-responder patients who don't show a decrease in size. But you can also see these great variability between patients. In some cases, patients you can see initially respond and then progress and so on and so forth. And so from these patient tumor burden trajectories from the model, they then were classified into 5 discrete states that were common trends in the data. And these traits include responder classifications in which patients either had a sustained response, as shown by this patient, where initially the tumor size was 100 millimeters and then decreased down to pretty much 0. So this patient was a great responder to the therapy. You have partial responders, which in this case, the patient initially started at 35 millimeters and went down to 25 millimeters. Then you have stable disease with no change, rebound disease where patients initially respond but then progress. And it's important to note that most of these rebound patients are found in the high-dose intermittent ribociclib arm, where they initially go down and then they go back up. And then lastly, we have progressive disease, those patients that do not respond to therapy at any time. So we classified patients into responders and non-responders. The next question was really to understand what the signaling traits between those populations were. In patients who are not responding, really what's the signaling changes in those tumors that are creating a resistant state. And to just describe our method briefly, again, from these women, we obtained the biopsies, which is represented by this little chart here. This is the biopsy. It's not true to size, but this is kind of what they look like. They come in this block. We then remove any OCT or paraffin, and then we chop it up and extract the nuclei. Each individual nuclei is in sequence, and so you get a transcriptional profile for each individual cell in the tumor. And we normally sequence a few thousand tumor cells for each patient in each time point. And so that really provides a rich landscape of phenotype and signaling dysregulation in these tumor cells over time. Again, you can also get from these same cells the whole genome sequencing. So our bioinformatic approach is just summarized here. Since this isn't bioinformatic talk, I'm not going in much detail, but I just wanted to highlight that, again, from the bulk tumor, we are able to isolate the signaling from each individual cell. You can imagine you might have some cells, for example, that have loss of estrogen receptor in these green cells, whereas these pink cells might have estrogen receptor expressed. And that's the type of data that the single cell RNA sequencing can provide. This data is then summarized by pathway. So pathway basically means all the genes in the pathway and if they're enriched in a cell or not. And this annotation is then used for the mathematical models or to inform experiments such as the organoid system that I'll be describing later on today. Okay, so shown here is this one of our patients. And each little dot represents an individual cell. And this is just plotted by UMAP algorithm, which describes cell states and their similarity. So you can imagine that in this figure on the right, what you're seeing is the pink are cancer cells and the blue are actually macrophage cells. And so we can, with the single cell data, annotate the cell classification. And for this presentation, we're focusing on the cancer cells. And if you look at the cancer cells, you can see that they cluster per time point. So these are the cells from day 0 from this tumor, in day 14, and day 180. And interesting, you can see that by the UMAP, you can see that they're quite distinct and potentially have distinct phenotypes across time as the patient tumor is being treated. Now we have many patients with this data, and I'm just showing this slide to indicate that this is just 21 of our patients in 63 time points. And the hot pink is the cancer cells. So you can see that for most tumors, we are able to obtain cancer cell annotation, but we also get a lot of normal cells, which are classified on the bottom here by color. And so each tumor has its own microenvironment, and the changes over time with treatment change also in that microenvironment. So this is an area of our research that's also ripe for collaboration and progress. But for this case, we're focusing on the cancer cells. And so Jason built a model for responders and non-responders that were classified by his imaging model to identify those signaling pathways that are different in the non-responders compared to the responders. And to do this, he built a hierarchical regression model that accounts for the variation between patients and within patients over time, as well as variability in the baseline expression. You can imagine, for example, that baseline in the patients for different factors might be higher than others, so they normalize the pathways to baseline. And then also account for changes in expression within individual patients. For example, you don't want one patient with a really high, for example, signaling pathway turned on to drive the discovery of common resistance pathways across the patients. So the top pathway was identified from Jason's model by using the non-responder-responder classification from the imaging data. And that pathway activity level is shown on the y-axis, and each line represents an individual patient in that particular arm, so each dotted line, I mean, and the solid line is the average of all the patients that were either non-responders or responders. And so what you can see is that in the letrozole alone standard of care arm, over time, so this is time 0, 14, and 180, you can see that the non-responders have more estrogen receptor-related signaling than the responders. So the drug letrozole is working effectively at blocking estrogen signaling in the responders, but in the non-responders, the estrogen signaling is still occurring. So they still are maintaining estrogen signal and therefore perhaps proliferation. Now in the combined arm with the letrozole plus either intermittent high dose ribociclib or the continuous low dose ribociclib, you see an interesting pattern in which the responders still have diminished estrogen signaling from the drug treatment, but the non-responders are losing all estrogen signaling. They're becoming estrogen independent. And so when you think about these, each of these, the non-responders in each of these arms, and you think about the signaling pathway, which is shown here, you can see that in the letrozole alone arm, you still have some estrogen signaling through ER, creating proliferation, and that's why they're still growing. However, the non-responders are completely bypassing this signaling pathway. They're no longer using this to grow. And if you look at the cell cycle activity, so this is just a pathway that summarizes cell cycle activity, again, in each of the different arms, you can see again that the responders have diminished overall cell cycle activity, whereas the non-responders have higher cell cycle activity. And in particular, in this intermittent high-dose ribociclib group, we see initially that these patients at day 14 are responding, but then they peak up and they have much higher proliferation at day 180. And this really reflects that trajectory we saw in the imaging data, where we saw patients who initially responded, but then rapidly became resistant with the duration of drug treatment. So with this model, we know that estrogen signaling is decreased, and the cells in the combined therapy arm are utilizing a different signaling pathway perhaps. And so we looked at the most significant pathways that are dysregulated in the resistant patients as compared to the responder patients. And we found that the junk pathway was maintained high levels of activity in the non-responders. So this plot shows on the top panel non-responder patients with either junk in the pink or ERK in the blue activity. I'll describe what these proteins are in a minute. And you can see that in the non-responders, junk remains really highly activated, whereas in the responders, ERK is maintained. And if you think about the MAP kinase pathway, which is the last signaling, I think, figure that I show, in this pathway, there are kind of a sequential activation of downstream kind of proteins, which then activates transcription through FOS and Jun. And this can occur through junk in specific upstream regulators, or ERK, which can interact with upstream MAP kinases as well as estrogen receptor. So in this case, activation of junk or maintenance of junk activity provides an alternative method to create a proliferative signal through FOS-Jun. So we believe that the working model for the signaling currently is that in responder patients, this pathway is effectively blocked by the either endocrine therapy or ribociclib. However, for those resistant patients that are progressing, that have alternative pathways activated that are endocrine independent, they're using alternative receptors and downstream pathway members to create a proliferative state. And so when we look at the single gene expression levels for these upstream and mid-level MAP kinase activators, they're also turned on in the resistant patients. So we have the pathway analysis. We believe we have a hypothesis for this, and we believe we understand that the cell cycle block can be bypassed through activation of this alternative pathway. So to further test this in a mathematical model, Jason built this model shown here. And in this model, he looks at actually 4 states, either estrogen dependent or independent, with a functioning or rewired cell cycle. To save time, we'll just be talking about the most impactful states shown here in the star, the estrogen dependent functional or estrogen independent rewired. Excuse me. And these are the predominant cells that we see in our patient tumors. Now, the model looks at how cell cycle progresses. So this figure represents a simplified cell cycle, similar to the earlier figure that I showed, in which the G1 checkpoint decision can either be blocked by competition or activated by estrogen itself or an alternative growth factor, such as activation of the junk pathway through upstream receptors, shown here, or it can be bypassed by activation of downstream pathways that are independent of estrogen, shown here. And so in this model, we then look at the role of the proliferation signal and block in either progression or quiescent cells. And so the color represents how much the alternative pathway is used, for example, the junk pathway. So yellow represents high, and the purple would represent low. And in this case, you can see that with an alternative utilization of an activation pathway shown here, we get higher levels of proliferation as compared to when you have low activation across estrogen growth factor utilization. So this really kind of highlights the importance of an alternative node to bypass the proliferation block. If you likewise look at quiescent cells, according to drug concentration, you can see again that with high alternative growth factor utilization, which is again in the yellow, you get lower levels of quiescent state and higher levels of proliferation. So since these data fit our expectations, we then move to the patient data. And so we really wanted to understand in part the role of these different CDKI drug doses and timing. So the pink line shown below is the endocrine plus low dose continuous ribociclib treatment. The green line is the endocrine therapy plus the intermittent high dose CDK inhibitor. And the blue line is the endocrine therapy alone. So for each panel, on the panel on the left, you see the estrogen dependent cells with the functioning cell cycle. These are the sensitive cells we've been talking about. The middle panel represents the estrogen independent rewired resistant cells. So these cells are the ones that aren't responding to the ribociclib and are progressing. In the model, and this is the total population, in the model you see with no therapy, those sensitive cells grow fast, right? They're outperforming, they're proliferating at high rates. Versus in endocrine therapy, their growth is impeded, and it's impeded further with the combination therapy. And this is normally what we'd expect for these cells. Now, with the estrogen independent rewired junk active cells, you can see here that the cells that dominate the growth in cold culture, of course, when the patients have these heterogeneous tumors, is the treatments that cause increased growth is the endocrine plus intermittent CDKI treatment. And so because it's intermittent, you see this kind of jagged kind of pattern that it follows. But you can see that the resistant cells grow much better with the intermittent kind of high dose therapy. And this might be, again, what we're seeing with that arm B with the patients that initially respond but then progress. So this model really fits what we're seeing in our actual patient data. And then lastly, this is the total, where again, you can see total population resistant cells increases over time with the intermittent high dose therapy. Excuse me. So intermittent high dose treatment accelerates growth of resistant clone development. And so we kind of feel like this story is coming full circle. So this is a summary of the current model is that based on the imaging data, we can classify patients as responders and non-responders. It's difficult to classify cohort of patients. And that this resistant disease is caused predominantly about bypass to the normal signaling mechanisms in breast cancer through an alternative pathway. And so because these are patient cells we're taking post-treatment, we need a system where we can really kind of test this model. So this leads to our model system in which we use ER-positive breast cancer cells in an in vitro system to really understand interaction between resistant and sensitive cells. So to build this model, and this initially was done by Vince in my lab, and Rena has since taken over this project. You take breast cancer cell lines that are initially sensitive to drugs such as ribociclib, and then you treat for a long time, months and months in culture, with either control, so they're not becoming resistant to anything, they're just dividing, or ribociclib. And so under this selective pressure, the cells gain resistant mechanisms. Surprisingly, I'm not showing this data, but they overlap almost exactly with the patient resistant mechanisms that we discussed earlier. And they become resistant to therapy. We then label these cells so we can see them in imaging. So the sensitive cells I'll be showing you are green, and the resistant cells are blue. And this really allows us to track their growth over time using imaging approaches. And so if you just kind of look at these cells and how they're growing, this is a reality check. And so on this panel, you see either untreated cells grown in co-culture, ribociclib treated, or estrogen. You can see that in the untreated, the sensitive cells have higher proliferation rates than resistance. So there is initially at least a fitness cost to the resistance. And then with ribociclib treatment, the sensitive cells no longer grow, right? They're not resistant to that treatment. So the resistant cells take over. And then with estrogen treatment, resistant cells don't really care. They don't really need estrogen that much. but the sensitive cells grow much better. So this model system accurately kind of recapitulates both the genetic resistant mechanisms as well as the growth traits of the patient samples. So the first question we asked was, because junk signaling is so central to the resistant cells in upstream receptor activation, would the resistant cells acquire sensitivity to drugs that target those pathways? And likewise, would then an ERK inhibitor be more effective in the sensitive population? So this shows a growth curve for either the resistant or sensitive cells grown in monoculture. So they're not yet grown in co-culture, just grown individually and at higher doses of either ribociclib, that's the control, or the ERK inhibitor, or upstream receptors of junk and ERK, and IQ3, which is a junk inhibitor. What you can see is that these cells perform according to our hypothesis, in which case the sensitive cells are more sensitive to the ERK inhibitor, and in contrast, the resistant cells are more sensitive to the receptor inhibitor and the junk inhibitor in this case. And so these pharmacologic results parallel kind of our model kind of pathway that we are working with. But tumor cells don't grow as monoclones, right? They grow in these heterogeneous populations. So really we wanted to see how these resistant and sensitive cells grow together. So this figure really kind of shows some of these results in which you see these are the resistant and sensitive cells grown either in monoculture, that's the far left plot in the green cells, either with untreated or ribociclib treatment. See, there's much fewer cells here because they're dying because of ribociclib. And on the right-hand side, you see monoculture, the resistant cells, which aren't as sensitive to the ribociclib. And then these three middle panels represent the co-culture system, where you have either both imaging kind of wavelengths shown here, or just the green or blue wavelength shown here. And you can clearly see that in co-culture, the sensitive cells grow much better than a monoculture. So what you normally would expect, because these are plated at 50-50 density as opposed to this, which is 100% sensitive cells, you normally would expect half the number of cells here as you see here. However, we see approximately 4 times that amount, and that's quantified at this bottom figure, in which the light green line represents the co-culture sensitive cell growth, and the dark green line represents the monoculture sensitive cell growth. And so we see initially right off the bat with this model system, we saw the expected sensitivities to the ribociclib, but we saw kind of a facilitative signal in the data, which facilitation basically is where one cell, such as the blue cell, helps the green cell grow under selective pressure, kind of shown here. But with the different growth rates of these cells and the different sensitivities to drug and so on and so forth, we actually wanted to further test this hypothesis of facilitation. So in that case, Fred built just a logistic regression model that accounted for the different growth rates, competition, and drug effect to see if indeed we're seeing facilitation. And so these results on the right represent, I think it's approximately 10 separate independent experiments with mono and co-culture that I just showed in the last slide, where the gray line from the model represents no cooperation or antagonism between the cells. And if cells are on this upper panel, and this is plotting just the sensitive cells, you can see that in co-culture under selective pressure, the cells have high rates of facilitation. Now, if we plot the red cells, they compete with the green cells, outcompete the red cells, but there's no facilitation or growth of red cells under selective pressure. So this is a specific finding to the sensitive cells. Okay, so I just have a couple more slides. In an independent model that Jason built, a life history model, he also found strong evidence of facilitation, and his model indicated that the sensitive cells are probably secreting a growth factor that increases their growth. So this model really informed our experimental design, which is now looking for the growth factors. And in this experiment, we took either the sensitive or resistant cells grown in monoculture or co-culture and performed liquid chromatography tandem mass spectrometry for estrone and estradiol. So estrone is the more inactive metabolite of estrogen, and estradiol is the highly active, potent metabolite. that activates estrogen receptor signaling and proliferation. And if you just look at the resistant cells, whether they're untreated or treated, you see higher levels of estradiol. So that's that blue line shown here. And in co-culture even, you do see again this high level, this not high, but significant increase in estradiol levels. And remember that the sensitive cells, just like our patients, are more driven to grow, to proliferate through estrogen signaling. So whereas the resistant cells are trying to activate low levels of estrogen signaling, so you see this higher levels of estradiol, it's the sensitive cells that can really act on that higher levels of estrogen and proliferate quickly with that particular signal. And so here this summarizes what we think is happening in these co-culture model systems, in which in co-culture under selective pressure, you have sufficient resistant cells present, which are secreting estradiol, which then serves to cause higher levels of proliferation of the sensitive cells under ribociclib treatment. And so in this case, there's facilitation of sensitive cell growth under selective pressure by the resistant cells. The last question we had, at least for this presentation, is what's the consequence of facilitation? When we started thinking about it, we didn't know, would facilitation help the patient or hurt the patient? And so to try to address this question, Fred extended his model to look at, to estimate growth and cell number with facilitation. So this model is a simple model that shows kind of under no facilitation, which are the dotted lines, what would the growth look like under selective pressure. You can see that the resistant cells would outcompete and basically take over the tumor. And however, with facilitation, which is the straight line right shown here, you can see that when you look at the population composition, there's both sensitive and resistant cells within this population that diminish the growth capacity of the tumor. And so we're building a more sophisticated model that accounts for continued evolution of the cancer cells and these interactions that we see, and to hopefully further delve into these models, as well as deriving further experiments to test the interaction of these cells in long-term culture conditions based on the models and the data that Jason and Fred provide. So in conclusion, hopefully what I've shown here is that the union of patient clinical and genomic data with mathematical models helped us to identify clinical response to therapy, the important resistant states across these patients' time and their treatment strategies, and interactions between tumor populations that may impact response to therapy. With that, I'd like to thank the team. I showed the folks that provided most of this work in that first slide, but we have a large amount of people working on this. And then during the questions, I just wanted to leave our questions up just in case anyone has answers. So these are the key questions that we're working from right now. So if you have any ideas or like to collaborate, my email is shown below. We're happy to work with others on these important questions. And that's it.
Okay, great, Andrea. That was beautiful and nicely timed too. So we've got plenty of time for some discussion. While you were presenting, there was a few Q&A that popped up. So some of those are related to the start of your talk. One in particular talks about how the biopsy really only gives local information, which may be very partial, especially in a heterogeneous tumor. How do you know that the whole tumor is represented by this biopsy alone?
So yeah, I keep telling the pathologist that I deserve the entire tumor, that they shouldn't get such a large chunk. But clinical practice is such that for this is estrogen receptor positive earlier stage, not metastatic. And so this is clinically what's ethical to provide to us. We have a separate project with autopsy patients in which we get a larger chunk of the tumor. It can look like heterogeneity within a tumor. And so we think the power from our study is really that we have 120 patients over the course of therapy. So it's not like there is some variation due to the biopsy, but hopefully that's countered by the numbers we have with the study and the numbers of single cells that we're looking at. It's always a risk though. And so we're complementing this study with the autopsy study to really better understand the heterogeneity across space. So that is the second part of our large grant. I just didn't show that work today. So that's a good question. I see the other questions. For intermittent ribo treatment, how were the dose strength and timing determined in the clinic? So the clinicians have a range. They know from a phase 1 clinical trial what doses they can give and what the standard timing is. And so they can divert slightly from that to try to understand the nuances of these features in their clinical trial. So for them, it was a PK/PD type phase 1 question that led them to the doses and timing that they had. So it was really kind of a clinical, it wasn't a modeling. It wasn't the modeling that had them decide this. It was really a clinical feature that they were capturing from the earlier studies from the phase 1 trials.
And then there's one there about proteomic analysis. Would that have given you more insights?
Yeah, you know, I'm always torn. I always want to do proteomic analysis, but the challenge is that with a single biopsy, you have to focus. You know, I don't have enough for every assay I'd like to do. I do think proteomic analysis would provide a level of understanding at the protein level what's going on. For example, we might see higher levels of the MAP kinase or JNK pathway, higher protein levels. Whether or not that would be consistent with or complementary to the transcriptional profiles, I'm sure it would help, but would it supersede the transcriptional profiles? That's always the type of decision I have to make because I don't have enough sample to do everything. So yes, I think it would help gain information, but in this case, since we have just a small biopsy, I have to prioritize the different studies tests that we do. Now with the HTAN network, I'm hoping that they develop really sophisticated processes that use really small pieces of tissue for all of these assays, so we can do more with a biopsy than what I'm currently doing. So if we had enough, I would absolutely do that.
Cool. Fred's made a little comment there saying you didn't emphasize this much, but if you use ribociclib in the Feline trial, it didn't have much of an impact. So what is the heavy selection pressure?
Yeah. So you saw with arm B that patients initially responded, but then they also progressed more rapidly, right? So because you're adding increased selective pressure on those patients, they're progressing, at least from what we can tell, more rapidly than the endocrine therapy alone. And so I think the response metric is confounded by these types of trajectories that the patients are having. And interestingly enough, there's a sister drug, abemaciclib, that hits different targets somewhat, and that drug was positive. So it might capture some of those resistant mechanisms, and so then lead to more effective combination therapy. So yeah, so that's our hypothesis. Thank you, Fred. That heavy selective pressure created an environment in which the patients evolved faster, tumors evolved faster, and created resistant state.
So there's a couple of questions there, and one of them relates to a question I want to ask, which was you talked about the intermittent dosing, but you never actually articulated what that intermittent dosing schedule was. And could there be a better way to deliver the intermittent dosing, like strength and timing, spacing, so on?
Yeah, so that's what I'm asking Fred and Jason to provide to me, the hypothesis, and then we'll test that in our model system. So absolutely, Sandy, we totally agree with you. There should be, but you know, when you start designing the experiment, there's literally thousands of approaches I could take. So this is where the modeling really comes handy to let me know what the important kind of timing and dosing I should focus on initially to kind of nail it down. The dosing in this experiment was 3 weeks on, 1 week off. So, you know, in breast cancer, the clinicians don't like to withhold therapy, even if it's an additional therapy. So it's kind of changing how they do things, right? The intermittent therapy, you get some hesitation. So yeah, in future subsequent studies, hopefully we can optimize the timing and dosing so that we don't create more resistance through the therapy.
Cool. Somebody's asking about, do you think drug resistance is a fixed state under your experimental time?
Is that your people, Sandy?
Yeah.
Yeah, you know, sometimes we see genetic alterations where, for example, like amplification of CDK6, for example, and I think that is a more fixed state than the transcriptional profiles. And so we see a little bit of both. But I think in those cases where there's a phenotypic switch, that probably isn't a fixed state and that we could reverse that state. And that does account for a lot of our patients. So it's a really interesting question, is the genetic resistant tumor cells, are those more fixed than the phenotypic resistant cells? And so I think that's something we'll have to look into.
Cool. And then there's something here about, they put it in the wrong path, but intermittent treatment was disappointing because your model oh no, you've already answered that. Combination therapies, we expect to use the second drug to act on the second pathway so that you hit the cells selected by the evolutionary bottleneck. So here the combination shouldn't be with drugs acting on the same GNK pathway.
Well, yeah, in the case of, so standard pharmacology is you get synergy when you effectively hit multiple nodes of the pathway and shut it down, right? So in the case of premenopausal, so it's a really interesting question. In the case of premenopausal women, I didn't say this, but this combination is effective in that population. Now, the estrogen signaling in postmenopausal women is different. And in this case, there's multiple clinical trials that failed in this population just recently. And so I think we need to better understand the role of estrogen and the endocrine signaling in postmenopausal women to better design therapies for this population of patients. I'm not sure if that answered the question, but there's some clinical reasons why I think this trial failed for this cohort. Remember, it was very successful for premenopausal women.
So then I have another question. If you just go back a slide,
yes,
because you had you flashed up a model.
Oh, that's
no,
wrong.
one more. That's forward, I think.
Wait, forward, back.
This, not forward, forward. You had it was the coupled system, and no, you went away past. It doesn't matter.
Okay.
But it was two equations, two ODEs, and it was clear that you had a sensitive and resistant population, and each of those had a carrying capacity, and it looked like their carrying capacity was different. And I recall Fred specifically making a point that, you know, why would the cost be in the carrying capacity? Why would such a thing exist? That's not the way to do it. And yet that's what's in that model. So I know Fred's listening, so I'd like to hear his response to that, but I wonder what yours is, Andrea.
I mean, I guess for me, I'm not convinced one way or the other, so I'm happy to try alternative methods to see what best reflects the patient data. So, I mean, we have some, you know, we have both the patient and the in vitro data. For me, I don't know what the answer is, but yeah, I'm happy to listen to you all to kind of address these questions.
Fair.
Fred?
Very diplomatic answer. Oh, it's a preliminary model. Don't worry, according to Fred.
Is that what he said? I don't know why we can't hear him.
The difference is through it's in the Q&A, the difference is through asymmetric competition, but we need a better understanding of what regulates the size of the tumor.
True.
Okay, let you off, Fred. Okay, I think Heiko has a question.
Yeah, thank you so much. Hey, Andrea, that was beautiful. Thank you so much. I very much enjoyed the visualization of your model fit to the data. When you had the longitudinal measurements, the red circles over time, and then the mathematical model with the bluish background of uncertainty given the imaging modality.
Yep.
And this looks to me like a fit after the fact, like a fit of the model to the data. My question is, if you just take the first few data points, would these models be able to predict forward in time how individual patients would flare?
Yeah. So that's a great question. And, you know, the interesting thing about the imaging data from the feline trials, it's pretty sparse. And we have 120 patients, but that's still sparse as well. So we are asking this question in a different context with ovarian cancer patients in collaboration with Peter MacCallum Institute, where they have hundreds of patients and they have less sparse CA-125 levels to measure kind of tumor trajectories of those biomarkers. And so in that context, Fred and Jason are working to kind of predict, extrapolate out from those measurements what's happening next. I think for the feline trial measurements, I'm not sure we can predict yet, because I think there's enough uncertainty and we don't have enough patients to really feel confident in taking that approach yet. But in the ovarian cancer, I hope that we can take that approach due to the increased number of patients that we're looking at there and the more consistent measuring of CA-125 over time.
Yeah, that makes sense. Thank you.
Okay, well, let me see. I think there is one last question there from an anonymous attendee. Do changes in the whole genome and single-cell RNA-seq under different arms help in classifying the responders and the non-responders?
So the question, Sandy, so the question is, does all the data, if we take an empiric biomarker of the single-cell RNA-seq data, is that better than the pathway level data?
I just think they're just saying basically, can you use the data to classify?
Yeah, so yes, yes, you can use you can develop an empiric biomarker from the single cell data to predict response to therapy. And we're doing this. My other postdoc, Vitro, now has a biomarker that utilizes this data holistically as an empiric. For us, for this project, we like summarizing the data at the signaling level so that we understand the biology driving the resistant state. So we love developing biomarkers, and yes, you can, and we have a few of those ongoing. But in this case, we summarize the data at the pathway level to really get to the root of the resistance mechanisms. So that's a good question. That's a good approach clinically to take to help these patients, because as you know, like they don't know who's responding or not. And so in 3 years, we'll understand the 50% of patients that progressed or recurred. And I think at that point, it'd be even more effective to build a biomarker based on that 3-year time point. Because right now we have like, did they respond acutely to therapy? But in 3 years, we'll know, did they respond, does those measurements that Jason built, did they reflect long-term recurrence or survival? So yes, that's a great question. I hope in the next few years to try to tackle that.
Cool. So you hinted at sort of in your some of your answers there that the second part, or the part still to come, is looking a lot more at the spatial aspects of some of these interactions. How far in are you on that? Because you're halfway through your CSPC, or what's your...
Are you a spy for the NIH, Cindy?
Not at all. I'm intrigued. You know, I care about space.
Yeah. We're doing an excellent job. We're awesome. We're totally in line. I think the spatial heterogeneity is a hard question, in part because we're using the autopsy patients. So every patient-based research is challenging due to various challenges with the tissue acquisition or processing. But the autopsy is particularly challenging. But we have been able to obtain single-cell RNA-seq data, and I don't think this has been published before on autopsy patients, for a subset of breast cancer patients. And so in a way, it's going well, but in another way, there's still challenges. Here we have 120 patients, and we feel confident in our results. But with the autopsy, we have 7 patients with viable single cells across multiple sites, and so we need to make sure we're powered and we have the right, we're cautious and we make the right conclusions from that data. And that's a little bit harder, I guess, than this particular case. So we're making good progress, but it has hit a few roadblocks that we want to make sure we're addressing before presenting that work.
I mean, it's a hard nut to crack. Since nobody else is going to ask, let me ask one final question. And it was when you showed right at the beginning your sort of TISNE patient data where you'd aggregated all time into one plot. What would be cool would be to see that over time. Now, I don't know if you can do that, but I presume from each sample you have a number of cells. But I don't know if the reason you aggregate them is because there's not enough data, or maybe you can articulate, but I would love to see the change in that over time.
Well, Sandy, I'm going to email you because we have the phylogeny of the single cell data for each individual patient in all their cells. And we overlay that with the resistant mechanisms. So I didn't show it for this presentation because I thought it might be a little much, but I'm happy to share that with you and get your take on it, because it is interesting to see how the single cells progress, the clonal populations progress over pseudotime or time, and how that interacts with the resistant mechanisms. And so I think what we're surprised about is that there's less, with resistant mechanisms, I thought within each individual tumor, there'd be multiple resistant mechanisms that are prevalent. But what I'm finding is that oftentimes there's, at least in this particular cancer patient population, there's oftentimes like loss of ESR1, upregulation of junk, and there you go. So it's not as complex, resistant, like heterogeneity, the resistant mechanisms, at least within this population, isn't as complex as, for example, our really late stage breast cancer patients. So it's been interesting to do that work, and I'm happy to work with people on that as well. It becomes a nightmare of figures in all the different kind of formats. But it's really interesting results from that data.
Yeah, I mean, that's the thing, is that ultimately you're becoming a bit of a data wrangler as well as a biologist and working with modelers, right? It's a complex interaction.
Well, and an artist. So I think our team is the worst.
There's definitely an art to presenting this type of result.
Yeah, we need an artist for our team. If there's any mathematician artists, please email me. We're happy to work with you in particular.
Cool. Well, thanks, Andrea. That was a lovely talk, and it was a great, I think, opportunity for the mathematical oncology subgroup to see some real kind of mathematical oncology in action in happening by a biologist. So from that point of view, it was very beneficial. So thank you, and I think you articulated the model parts of it very well. Admittedly, they were nice interacting visuals and not the equations, but I still think you explained it nicely. So thanks for that.
Thank you, Sandy, everyone.
Yep. So for those of you that are interested, don't forget we have our Bring Your Own Beer happy hour in just a few minutes on the mathematical oncology floor. There's a Bring Your Own Beer room. We'll be there drinking and celebrating Andrea's great talk. And also, if you have a few dollars left, don't forget to go to the store and buy yourself one of these funky t-shirts. Okay. Thank you. Thanks, Andrea.
Thank you. See you.
Bye, everyone.
Bye.